talk about two things. First, our current test as it relates to the surgical procedure, but also our next generation platform and the progress and the plans we have there. And I think it's interesting to pause and take a look at where is cataract surgery in general. And if you think about the amount and the pace of innovation, it's truly incredible. I think it's evidenced by the presentations, many of them you've seen this morning. If you look at today's procedure, it probably would bear little resemblance to the procedure 10 years ago. And it's gotten to the point where the ocular surface has really become a key rate-limiting step in achieving your surgical outcome. And it's no surprise when you think about all the different ways the ocular surface plays a role. We know that it can impact IOL calculations, the quality of the ablation patterns, how a patient heals, and ultimately, how well does that patient see? And I think it's important also to pause and realize that once you begin surgery, the physiologic state of the eye is altered. And there's nothing you can do during surgery to account for that specific patient's basal tear film properties. It's important to have that information beforehand. And that's important because sometimes it's easy to tell when there's going to be a problem, but sometimes it's not. No one has a problem diagnosing a severe patient. Everyone knows that patient's ocular surface disease has to be addressed before proceeding. But the mild and moderate patient can be equally problematic for a surgical procedure, but much more difficult to diagnose because many times they present very similarly to normal patients. And that's where testing for osmolarity can really play a role. And what you're looking at here, these images are at a 200x magnification, and they're of human corneal epithelial cells. So this is a view you're not going to get at the slit lamp. And where I'd like to point your attention is to that middle image. And what you can see is when these cells are exposed to a level of hyperosmolarity that's consistent with what a mild or moderate patient would experience, even there, the patient is having significant apoptosis, which translates to significant tear film instability. And these are patients that are often missed. So how does that play a role in cataract surgery? Well, here's a study that was done by Dr. Epitropoulos, and what they did was looked at the average K values between two visits. And what they found was when patients had normal levels of osmolarity, the readings were very consistent and very similar, as evidenced by the tight grouping on those blue dots. However, when the patients were hyperosmolar, 17% of the time, those readings had a greater than 1 diopter difference. And there's two important points here. If you look, this was consistent, not just in the severe group, but also in the mild and moderate. And these patients, 2/3 of the time, they required having their IOL power changed before proceeding to surgery. So we know osmolarity is important, but we also know it's not the only biomarker. The tear film has a tremendous amount of information in it. And so as we go forward, where we'll be looking initially is at markers related to ocular surface disease. And just a few examples are IL-1RA, which is an excellent marker for inflammation. I think everyone's familiar with IgE and its role in allergy. And even something like lactoferrin is interesting because it can be used not only to subtype dry eye disease, but early data are showing that it's an interesting marker to predict postoperative healing dynamics. So there's a lot of exciting information available in the tear film. So where are we with our platform? Well, we believe that it will continue to be important to have a quantitative result very quickly. And we're going to add that, the ability to multiplex multiple markers on a single test. So doctors can differentially diagnose, they can subtype disease, and they can direct therapy. In addition, when we add on the fact that we'll be capable of EHR integration, we think we'll continue to be the ideal solution at point of care. From a research standpoint, we'll also be able to normalize using osmolarity, which basically means it's going to render other biomarkers you measure in the tear film very informative and a lot more accurate. And finally, we'll be able to customize with intentionally designed sensitivity and specificity. So where are we in the process? We're on pace to have CE mark by the end of the year. We will have our 510(k) submission in early 2017. And our plans up right now are that we'll have 1 to 2 additional markers with our next test. All of our tests will always have osmolarity. Our next test is likely to be focused on inflammation, but what's important is that we truly have a platform, and we're going to be able to bring out new iterations every 6 months. And where we go will be determined by where's the greatest need and where can we add the most value. So in closing, I'd just like to say how excited we are at TearLab to be pioneering the future of tear film diagnostics, and we know that together we can absolutely elevate patient care. Thank you.